Okay, here we are. This is our tutorial 3. Right now we're doing question 1, and let's just get into it. So, it's saying we're studying an operon, a species of bacteria that contains all 3 enzyme coding genes of the so-called product metabolic pathway, and then another gene X encodes for regulatory protein. So first we're going to draw and label the operon. So this is what our sequence should look like. We have our gene, X gene, that encodes for regulatory protein. We'll have our operon, and then we'll have our A, B, and C pathway, which code for this metabolic pathway. And then what happens is when X encodes for this regulatory protein, it'll bind to O. And it will, when it's bound to O, it keeps the operon off. So when RNA polymerase tries to come in, it blocks it, and A, B, and C can't be metabolized. So this is very similar to a lac operon. So let's see what B says. It only is produced at high levels when a rare amino acid becomes abundant in the growth media, and this amino acid represses the regulatory protein. What type of operon are we dealing with? So this is very similar to a lac-out operon, which is inducible, because what happens is this rare amino acid, here I have it here, you can kind of think of it as lactose or allolactose, because when it's available, it binds to X, and that releases this it binds to this regulatory protein and releases from the operon. So here I wrote rare amino acids, kind of similar to lactose or allolactose, because allolactose suppresses the suppressor, so to say. So, yep, inducible. Let's check out C. We have given the answer to B, where might mutations that cause the operon gene to be constitutively expressed. Okay, so here I wrote we could have X minus is a mutation in the X coding gene, which makes it not function, and therefore the regulatory protein is unable to bind to O, which would keep it, which would turn on O and let the pathway metabolize. That is, of course, if A, B, and C are all A plus, the wild type, and they're able to be metabolized. Or we could have OC, which is a mutation in the operator, which is dominant to XS, X plus, X minus. It's the most dominant mutation. So in this case, it keeps the operator turned on, and it will be constitutively expressed. Okay, so now we're looking at the regulatory protein X has 2 binding sites, one DNA binding site and the other allosteric. If a mutation occurs in the allosteric site, what effects do you think will have on the accumulation of the pathway when the rare amino acid is abundant? So this is an example of this XS. Sorry, I'm a little shaky right now. This XS mutation, which is similar to the IS mutation in the, let me fix this. The IS mutation in the lac operon, but in this case it'll be XS. And XS is dominant to the wild type. So basically what happens is this rare amino acid is unable to bind to the regulatory protein's binding site. Alright, so yeah, basically what happens is the amino acid cannot bind at the allosteric site because of this mutation, which basically means that this regulatory protein will be stuck on O, turning off the operon, and the metabolic pathway just will never occur. Okay, so if that makes sense. And then E, if the bacteria of interest has 2 copies of the product operon and 2 copies of the X gene, do you think the mutation described in D would be dominant in the wild type gene, X allele, or recessive? Well, this basically just comes down to knowing whether or not it's dominant, and as we learned in recitation, the XS or IS is dominant to the wild type. So if let's say we had something like this. My pencil keeps breaking. If we had something like this, XS O plus A plus B plus C plus. These are all functioning, but this will never allow the operon to be turned on. And it was over here. Let's say this is X plus O plus A plus B plus C plus. This is always dominant over this, so yeah, the metabolic pathway will just never occur. Now, we have F. Imagine the bacteria of interest had 2 copies of the product operon and 2 copies of the X gene, with one X gene being upstream to the chromosome operon and another X gene being upstream to the plasmid operon. The genotype is the following, where plus refers to the wild type and minus is loss of function. So here what we have X plus, which is also dominant to X minus. We know that just through our studies. But in this case, even though the X plus is working, one, we have a lack of, where is it? Oh yeah, here it is. In the absence of the rare amino acids. We don't even have the rare amino acid, so X plus can't be, the suppressor cannot be suppressed, and we have A minus, B minus, and C minus, which would mean that the metabolic pathway just wouldn't occur anyways, even if there was that available. So here we look at the dominance between these two. We know that X plus is dominant to X minus, and that answers question F. And lastly, propose what if. So here, I just decided to say, what if this X minus in this hypothetical situation, which isn't possible, but let's say it is, that X minus is dominant to X plus, how would the outcome of this turn out? Like, would we get our metabolic product? And yes, since it's X minus, the regulatory protein is, or it's not even coded, so the operon stays on, and we have A plus B plus C plus, so there's no protein blocking or turning off the operon, so the operon's going to stay on, and we have A plus, B plus, and C plus, which are all functioning pathways to make our product. So yeah, it wouldn't happen, but if that was the case, that would be the answer. Alright, hopefully that makes sense, and catch you later.